Under the topic of blood, we are already done with number 1, the formation of blood as the first topic, number 2, composition of blood as the second topic, and number 3, red blood cells as the third topic. Now it's the turn for fourth topic, and that's your leukocytes. Leukocyte is the second formed elements out of the 3. So in the composition of blood, we saw that blood is composed of 55% plasma, and the remaining 45% includes the formed elements or the blood cells or the blood corpuscles, out of which the highest percentage is of red blood cells, which is 44%, and the remaining 1% is white blood cells and platelets. Among these 3 formed elements, in this particular video, we're going to complete the basic introduction and properties of white blood cells. So, the fourth topic is leukocyte. Let's break the word. Leuko means white and cyte means cells. Then obviously, white blood cells are leukocytes, or leukocytes are the white blood cells. What we are going to do is study the properties of leukocyte in a similar manner in the similar manner to what we did with red blood cells or erythrocyte. That is, first beginning with formation. So the formation of leukocyte in human body is known as leukopoiesis. So leuko is referring to the white blood cell, and poiesis means formation. So the formation of white blood cells in human body is known as leukopoiesis. Obviously, leukopoiesis takes place from leukopoietic tissue, or the tissue that gives rise to white blood cells. And these tissue or part, they are red bone marrow as well as lymph nodes. You don't need to know the process. All you need to know is that in human body, at red bone marrow and at lymph nodes, the process of formation of WBC takes place. Number 2 is the shape. So WBC can be amoeboid in shape, they can be rounded, or they can even be irregular in shape. The shape depends upon the type of WBC that we are referring to. And if you want to know what's the different types of WBC, you gotta wait for the next video. Thirdly, WBC count. The number of WBC found in human body ranges from 5,000 to 10,000 per cubic millimeter. 1 cubic centimeter means 1 milliliter. So 1 cubic millimeter, you can calculate it on your own. Number 4 is abnormal condition. When the number of WBC is less than the normal WBC count, that is 5,000 to 10,000 per cubic milliliter, then that particular condition, abnormal condition, is known as leukopenia. Whenever you see penia, then that refers to less in number. That's just one way of remembering it. Because if the RBC count is less than normal, or erythrocyte count is less than normal, then you would say it's the condition or abnormal condition of erythropenia. Similarly, when WBC count is less than normal, then that abnormal condition is leukopenia. If the normal count is more than if the count is more than the normal WBC count, then that particular condition is leukocytosis. When you see the part cytosis, you could remember it as being more, because when WBC count is more than normal, it's leukocytosis. When platelet or thrombocyte count is more than normal, then that's thrombocytosis. The same way leukocytosis exists. But that's not the only condition. The second condition is leukemia. Leukemia. These two conditions, they take place at two different conditions. One, leukocytosis. Whenever there is infection in your body, in order to fight against that infection, our body increases the formation of white blood cells. At that particular time, the number of white blood cells in your body becomes more than the normal count, and that particular condition is known as leukocytosis. So leukocytosis occurs due to infection, while on the other hand, leukemia is blood cancer. That is when your WBC starts to divide uncontrollably, then that's your leukemia. So when there's infection and WBC count increases, that's leukocytosis. But when your WBC start to divide not under control and you get blood cancer, that particular condition is leukemia. Average lifespan, it's from 8 to 10 days. Another important thing that you need to know is diapedesis. Diapedesis is a sort of action. What type of action is it? It's the squeezing action or squeezing out of WBCs from capillaries. In the diagram, you can see a capillary. Inside this capillary, there are WBCs. Through the thin wall of this capillary, the WBCs can squeeze itself out. Whenever, suppose there's a pathogen nearby, in order to eat, engulf, and destroy that pathogen, the WBC from the capillary squeezes out. And that particular process of squeezing out of WBC from capillary wall is known as diapedesis. Not only that, the addition of WBC within the lymph takes place through the process of diapedesis as well, which you'll come to know under the topic of lymph. Second is pigment. No pigment is found. That means there's no hemoglobin. Then function. WBCs, they are the soldiers of body. That is, they form our immune system. They are there to protect our body from harmful pathogens. Finally, nucleus. Depending on the type of WBC, the nucleus is different as well. So different types of WBC, different types of nucleus are found in WBC. And that's it for WBC. In the next video, you'll see the various types of WBCs, and that particular video is going to be much more interesting than this one. But remember all the points that I have mentioned here and compare these points with the points that I have mentioned under the topic of RBC. Bye.